Okay. All right. Thanks for sticking with me, guys. This is the last video, and we'll kind of go over the summary conclusions as well as some of the limitations and questions and my proposed experiment, and then we'll wrap it up with some future directions. So for the summary conclusions, keep in mind that they used a direct side-by-side comparison of the cardiomyocytes, progenitors, mononuclear cells, and even the non-cardiac group, which I forgot to include here, but that just served as their negative control to make sure that there was a control on the experiment. Um, so some of the highlights, um, they were able to characterize and demonstrate the three cellular sources that they were aiming for, which were the progenitors, the cardiomyocytes, and the mononuclear cells, and the negative control group. And by characterizing them, they made sure that the cells were the ones that they wanted. And then lastly, they analyzed function and performance for each cell group using different techniques such as echocardiography, histology, immunostaining for revascularization, their blood flow analysis with the microspheres, and with each of these results, they were able to make their own comparisons. So the researchers claimed that there was a superiority of the ES cardiac derivatives over the mononuclear cells for improving cardiac function 28 days or about a month after transplantation. And taking together all of their results, they stated that they support continued research in the use of cardiomyocytes derived from pluripotent human stem cells for heart repair or regeneration. And so, that was kind of the goal of their work, and it kind of helped address their hypothesis, which was, their hypothesis was that the progenitor cells originally were going to be the best, but, they found out that, hey, the cardiomyocytes could also be viable as well as an alternative to this gold standard mononuclear cell. So these were the most important conclusions from that article. However, there's still some limitations and unaddressed questions as well. The researchers did do a great job at designing their experiments and trying to show superiority of the cardiomyocytes and the progenitor cells. But when you look closely at the data, there's still a few questions and a few limitations. For one, their echocardiography results, remember with the fractional shortening and these two dimensions that were taken, they weren't actually absolutely convincing. And more importantly, after doing some further research, this fractional shortening can depend on a lot of other factors, including the geometry of the heart and its normality. So even if the heart is slightly abnormal or slightly not to the normal geometry, this can really impact those results and mislead a lot of researchers as well, which is kind of a big talk in the cardiac community, I guess, that's studying all of these factors. Next up, when you look at the histology evaluation, none of the groups really reduced the infarct size, which is kind of what we saw with that picture of the scarring and then their graph that was next to it that showed no significant differences between the non-cardiac group and all three of the other cellular sources. So this wasn't, entirely convincing either. And lastly, when you look at the revascularization immunostaining results, the capillary density didn't change aside from the non-infarcted area. So capillary density inside the graft and inside infarcted areas wasn't significantly changed by any of the three cellular sources. The only change that was seen was in a non-infarcted area, which again leads you to possibly conclude there's some kind of hidden mechanism. So the major question is, what is this mechanism? What mechanism causes cardiomyocytes or progenitors to be superior over the mononuclear cells? So my suggested additional experiment is to use the same experimental groups that they had, but perform different experiments to evaluate cardiac function and the performance of these cellular sources. So yeah, for instance, in their echocardiography, their results were kind of promising, but even with ultrasound, you can only get, oh, sorry about that. Even with ultrasound, you can only get a certain level of sensitivity and specificity with that kind of imaging. It's great for, motion imaging, which is kind of what the heart will require. But there could be other imaging and applications that could be done, or modalities that can be used to give greater analysis. So my first one is to test myocardial viability and use radionuclide labeling with PET and SPECT imaging. This could be done to give, and I'll explain it in the following slide, but this could help them, find that mechanism. They could also perform gene expression analysis with quantitative RT-PCR and DNA microarray analysis. Or they could use this iron particle labeling in an ex vivo approach with MRI. Or they could use reporter gene labeling in fluorescence/bioluminescence imaging. And this could be done to kind of better see how the cells are developing within the heart, so that they don't have to limit themselves to looking only at a baseline and only at 1 month after transplantation. They could perform this last technique, and possibly some of these other techniques as well, at different time points to truly see progression of the cellular source and see, kind of get more resolution at what changes are happening. So the controls for this experiment are the same ones, where the positive control is the mononuclear cells, and the negative control would be this non-cardiac group. And again, I included this article because it kind of highlights all of these and many other kind of evaluations that can be done with imaging or stem cell techniques to evaluate, how these cellular sources actually impact organs. And in this article, they do a great job of talking about different ways they could be used for the heart as well. So the expected results are that the myocardial viability, which is basically the contractile function with PET and SPECT imaging, that the progenitor cells and cardiomyocytes will have positive results, while the mononuclear cells will not have as improved results. And you kind of saw that with the echocardiography imaging. But with using these two imaging modalities, you might be able to get an even greater significant difference between the progenitor cells and the cardiomyocytes and the mononuclear cells. With gene expression analysis with qRT-PCR and DNA microarrays, I believe that the progenitor cells will demonstrate, the mesodermal regulators, which are common because these progenitor cells come from the mesoderm. And the cardiomyocytes should express cardiac-specific structural genes because they're differentiated from the progenitor cells and they're a little bit further along in their development. Next up, for the iron particle labeling with an ex vivo approach in MRI, you should be able to better see location of the cells in the heart. They've been used in stroke modeling applications, and with using this kind of technique, you could definitely see location and kind of be able to tell, especially with that revascularization question of why that occurs outside of infarct areas and not even in the graft as well. And lastly, they have reporter gene labeling and fluorescence/bioluminescence imaging. And this can help better monitor, progression of the cells as well. It can help monitor survival, migration, proliferation, and with all of these results, I think they could find, the true mechanism for why these cells would be better. My feeling is that, that revascularization does occur outside of the infarct area, and that nobody's really discovered that yet, but it's still kind of left up in the air due to their prior experiments. So some future directions. One is they can move to a larger animal model. That same group, I found out after doing some research, has already done some studies with guinea pigs and macaque hearts, and they've done some injury models that demonstrate cardiomyocyte usability. They still need to demonstrate that electromechanical coupling that I've talked about before. Perhaps they could also induce different types of infarctions to the heart, or in different areas of the heart they can induce those infarctions. So those two might be interesting ways to see, how different areas or different classifications could really impact their work, and how their work could impact treatment of those kinds of diseases. Uh, there's also They were also limited in this study to xenotransplantation models. That kind of limits their learning of how immune responses would evolve over time. And yeah, speaking on that point, their time frame was limited to the first 28 days after cell transplantation. So it could be shown that cardiac improvement at 1 month could not be sustained or reassessed at 3 months. So it could be important to kind of maybe lengthen that or at least get better resolution in their results. And the last thing they could look at is direct injection versus intracoronary injection. So that's it for my presentation. Thank you guys for listening. Um, and I'll be waiting for your comments to respond. Thank you.